Hi all, welcome back to Environmental Science, and we're going to continue talking about population ecology. Now we're going to talk a little bit about abundance, diversity, species richness, species evenness. It's going to be a recap kind of review of what we've talked about in class during class activities, but just want to make sure that you guys are familiar with this because it really plays an integral part in determining how populations or communities are changing over time. Okay? And so let's dive into it. So the abundance of, or the abundance in a population is the total number of organisms in either that community or that population. So remember, community is all the different species within a given region. Population is a single species and the individuals that reside or belong to that species. Okay? Diversity is the number of different species for a given ecological niche. Okay, and we can further develop that by calling it genetic variation. But I want to just want you guys to really just think of from an ecology, environmental science perspective, that it's the number of different species and kind of how they're evenly distributed. Genetic variation plays a really big role, but we're not going to dive into genetic variation in this course, because genetic variation, it gets at ways at which maybe the population is high for a given group of organisms, but due to something that we call genetic bottlenecking. Okay, so we talked about this just a little bit quite a while ago when we were talking about devil's hole pupfish. Okay, so we said that devil's hole pupfish, their population was really fairly large for a small body of water. So the population was like 400 individuals. And then ecosystem change happened and the population started to shrink. And then other things happened, human error happened, and the population really shrank. And it got down from like 400 individuals to about 30 individuals. Okay? And so you can think of kind of like a pop bottle. And so all those individuals went through this bottleneck. The individuals that survived, right? The 30 individuals went through this bottleneck. And now, as that population might grow again, the amount of genetic variation that you see is limited because only a few organisms made it through the bottleneck. Okay? That's where genetic variation plays in diversity. Okay, so even though you might have really high numbers in a population, if they're all the same individual or if they're cousins of each other, then the genetic diversity is still very low. And we can come back and talk about this later if this subject really intrigues you. But save it for other courses that maybe get into DNA analysis, genetic variation, population genetics, things like that. Okay? Abundance of particular species, okay, is often relates to the diversity. So remember, we talked about diversity as being both the number of different species and then the abundance, how many of that particular species are available. Okay, so that gets into species richness, and we'll come back and talk about this. That's the number of species, different species. And species evenness is the abundance. Okay? So as a general rule, diversity decreases and abundance within a species increases when we're moving from the equator to the poles. So if we throw this map out, this shows what we call species diversity. And you can see this is from a terrestrial perspective. Marine perspectives are a little different. It's more based on the currents and where the currents are going, and they change. They might alter that just a little bit. So you can get huge species diversity a little bit further up in marine habitats and a little bit further down. You can also get a pretty big band at the equator, but terrestrial habitats are driven really only by primary producers. That's what the diversity is driven, which is driven by that conversion of solar energy into chemical energy. So taking photosynthesis, converting it into chemicals, converting that energy that's in chemicals into body mass, variety, fruiting bodies, etc., leaf matter, etc., etc. That is primary productivity, and it's really, really high near the equator of the planet. You can see it all the way across because those regions are getting the most solar energy. As you move away from the equator towards the poles, your genetic diversity tends to decrease. Well, your genetic diversity, but also your species diversity tends to decrease as you move away from the poles because you have less plant. This gets back at what we talked about before, where we were talking about the number or the amount of energy that's coming out of primary producers directly relates to the amount of energy at the next level. Okay? And so if you have huge amounts of energy in primary productivity, then you'll have more energy in primary consumers and more energy in secondary consumers, and on and on and on. Okay? So another example of this is when we can look at different species and how that relates to different species. So we can look at things like frogs, and we can look at frog diversity. And frog diversity is high at the equator, and as you move away from the equator, you get less and less diversity till you really don't find any amphibians. Okay. We can look at mammal diversity. They're a little more suited to quite a few more environments, so they fill ecological niches, so especially rodents. But rodents are very similar, so fairly high diversity near the equator, and as you move away from the equator, less diversity. Okay? But still more diversity than, say, amphibians. This right here is probably the result of endothermic organisms that regulate their body temperature by consuming food and kind of maintain a constant body temperature, or ectothermic individuals that are their body temperature is the result of the climate that they're in. So ectotherms typically can't reside in cooler climates. So when you get down to the tip of South America, there's a lot of cooler climate there, and they really struggle with it. Bats, same deal. Birds, same kind of deal. So all these species, same thing. As you move away from the equator, the species diversity decreases. Okay, so those equator regions or equatorial regions are species rich. They have lots of different species, high species diversity, because high primary producer diversity. Okay. So let's get into that diversity calculation just so we can talk about it one more time. Again, diversity is often calculated using species richness, so how many different species there are, and species evenness, how many individuals of each species are present. Okay? I showed you guys Simpson's diversity index. Okay? There are others, Shannon Weiner diversity index. There are others that can get at a index of how many species or how diverse a ecosystem is. Okay? They're nice. They're not the end-all be-all kind of situation, but they're nice to see what trends are over time, or how two ecosystems or two regions might relate to each other, or might compare to each other. Maybe we have a region which has high human activity, and a region that has less humans. And we might look, well, is the diversity, is there a diversity difference, okay, based on species richness, species evenness? Okay, so again, remember the equation. Some of you are well adapted to running this equation, others not so much. You will see it again, okay, but just so you can kind of go through and check that out. Let's say we sample two populations. Okay, population A might be a pristine stream, but may have a high level of human exposure, meaning that humans might live on the stream. The stream is fairly pristine in the sense, but humans have replaced a lot of native primary producers with maybe like apple trees, or maybe they replaced it with trees that don't drop as many leaves, so conifer trees, and then maybe we have a native population that has less impact by humans. Okay, in first glance, we might remember we said before, when we did this in class, we said mayflies, stoneflies. Okay, these are great. These are great species to see. They're normally very intolerant of pollution. Okay, and when you see huge numbers of mayflies, like often you're like, hey, this is a great stream, and both streams are pristine. Okay, I've already stated that they're nice, clean water. Okay, very little pollution. Okay, but you have lots of mayflies in population A, and then a smaller representative stoneflies, dragonflies, craneflies. Population B, it's fairly uniform. Okay, so you have the same number of species. So species richness is equal, but species evenness is not. And so when we do the math and we look at this, okay, we can take and do that equation. So remember, little n is the number of individuals within a species. Okay, and so when you do that math, 52 times 51. 2,652, and we just go through. We can add that numerator, okay, together, and we can then do the same for the denominator. Denominator is the number of species, okay, added or number of individuals that have been collected, added together. So in this case, for population A, it's 68. So 52,259, 68. And then you take that times itself minus 1, so 68 times 67, so 4,556. And then when we look at that population, we have the species diversity of population A is 0.4, and the species diversity of population B is 0.76. Okay, so it really plays an important role to look at evenness, because even though they have the same number of species, okay, how they are evenly distributed across those species really dictates how well that diversity, or how well that ecosystem is doing. Okay, community structure. So now we want to talk about ecological structure. So we can look at ecological structure in two kind of ways. We can look at it from what we just did, species diversity. Okay, so we can look at how many different species you have in an ecosystem. How evenly distributed are they? Okay. We can also look at how the populations of different organisms are structured within the ecosystem. So do you have a lot of randomly distributed populations? Okay? Or do you have a lot of clumping of populations around a single resource? Okay? And so we can get at that by looking at distribution of organisms, of different populations within that ecosystem. So random distribution would mean that you have just the populations are just randomly kind of laid out on the landscape. There's not a heavy amount of competition. They're not seeking out a single resource. They're just randomly distributed across the population. So, like, we have an example of that here. So, like wildflowers that pop up in a meadow. Often those wildflowers, like here, you can see Indian paintbrush. You can see species of lupin. Like, when they pop up, they just pop up, and there might be no rhyme or reason where they're at. There's not a lot of competition. These are all typically, a lot of wildflowers in the sense that have this kind of pattern are what we call annuals. And so they produce a flower, they get pollinated, they produce lots of seeds, spit them out, and then the plant dies. Okay? And so they only live for 1 year. And then, if conditions are right, the next year, boom, you get another wildflower explosion. And they're not really seeking out nutrients or pockets of nutrients or anything like that. And there's not a huge amount of competition between the organisms. And so you can get this random distribution. Clustered or clump distribution, okay? Often you can get that around single resources. And so, like, maybe coral reefs are a really good example of this. Wherever there might be coral, you might have fish that are clumped around that because maybe they're feeding on a resource that's related to the coral. Or they might be using the coral as a place to deposit eggs, or they might be using it as a shelter to hide. And so they're clustered around certain resources. We can see this on landscapes where you might have pockets of trees. The trees might be clustered in water landscapes. So if you go to arid regions, deserts, or semi-deserts, you might have, streams and pools of water that happen on the landscape, and you have pockets of trees that occur along that waterway. Well, anything that is associated with those trees, nesting birds, that kind of thing, they might be clumped with that primary producer. And so that's a clustered or clump distribution. And then you can have uniform distribution, and most of the time uniform distribution is directly related to competition. So here you can see these nesting birds. You could take a measurement between these nests, and these nests are basically just maybe 2-3 centimeters longer than the reach of the bill. So they nest a specific distance away from each other, so they can't attack each other and reach and touch each other. Okay, but they nest close enough that they all can be on a nest, all in the same region, but the competition is for space. So they pack in as many individuals as possible, but it's a uniform distribution. You can see this in the desert with plants. So if you've ever been to a desert landscape, and you might say like creosote plants or sagebrush plants, and you might be like, wow, there's like a perfect path. You can walk in between all these plants, never touching them. That's because these plants are competing for water, and their roots, are competing underground. And so they basically establish their own little zone, and that's their growth zone, and that's the competition. And so you get these, evenly or uniform distributions of organisms across the landscape. So why does that matter from a perspective of environmental science or ecological management? Well, knowing especially things like that are clumped around a single resource, if that resource goes away, if that coral reef is damaged or is removed, or if that pocket of trees in a region is removed, then you can lose an entire ecological assembly. Okay? If the uniform distribution, if something disturbs that uniform distribution, okay, so if for some reason these organisms can't be uniformly distributed on a nesting ground, or if your water patterns are so much that the competition between sagebrush, it's still not enough to get water, then you'll see those patterns, those distributions change over time, and you might have less individuals, right, that can be established in a region, or you might have a change in the distribution. Okay? And the other thing that we often don't think about too often, because we're terrestrial organisms, so we often don't associate distribution in a vertical matter, okay? But in marine habitats, for sure, there's this vertical distribution. And so that vertical distribution, it could be random, it could be clustered, it could be uniform, depends on the ecosystem, but it can happen in a vertical zone. It also happens in a vertical zone in like trees and things like that, for organisms that might use trees as their ecological niche. Okay? But as you know, as terrestrial organisms, we typically don't think about that pattern, but it can happen from a vertical standpoint, not just a horizontal standpoint. All right, the next thing that I want to talk about real briefly is complexity, the complexity or connectedness of trophic levels. Okay, we've talked about this a little bit before, that diversity relates directly to the complexity of kind of your food pyramid, or your trophic levels, your primary producer, secondary, or primary consumer, secondary, tertiary, etc. And so that complexity, or the number of trophic levels, is often determined basically by that primary producer. Is there enough energy at the primary productive level to support multiple trophic levels above it? Or is there so little energy coming from the primary producer that maybe you can only support primary consumers, and there's just not enough energy resources? So how many trophic levels is normally directly related to how much energy is coming off the primary producer. So when we see huge ecosystem food webs that are related to things like krill in marine environments, or related to species in a rainforest, and we see all this connectedness, normally it's related to organisms, a primary producer that is very energetically abundant. So meaning that they're occurring in regions where there's lots and lots of energy that is being transferred up that trophic zone, or up to that trophic levels. Okay. So we've talked about it before. Okay. Again, primary producers, normally we call those, anything that's a primary producer, we call it an autotroph. Okay, so they produce their own food, and then anything above that primary producer that would consume it is called a heterotroph. Okay, even if they're just consuming dead material, we still give them the term heterotrophs. We call them detritivores, so worms, mushrooms, or fungi, other invertebrates like millipedes and some centipedes that would eat detritus, decaying material. They're detritivores, but they're still heterotrophs. Okay, and so that connectedness is really important from the primary production point of view, even driving the detritivores. So that's a key component. So how much dead, decaying material is still related to how many primary producers you have. Okay? And that really gets at these giant food webs that we've talked about before. And from a trophic level, the connectiveness, or the amount of connections you have and the amount of species you have and how evenly distributed they are within an ecosystem is directly related to how much energy is coming from the primary producers. Okay, so we're going to stop there. Next time we're going to talk about some stability of an ecosystem and what happens when we have ecological change, and maybe some information about resilience. Like, there are some ecosystems that you can remove primary producers and new primary producers can come in and the energetics really doesn't change. And then other ecosystems, you remove some of the primary producers and the whole trophic level just basically collapses. So we'll talk about that next time.